Hi, and welcome to this second lesson of the week. For you guys in year 13, this is the action potential, which is how an electrical impulse is transmitted down a neuron. So to start with, I just want you to answer these three questions. What is the value of resting potential? Does maintaining resting potential require energy? And will a generator potential always trigger an action potential? Pause the video and write a sentence or two for each of these answers. Off you go. Okay, so let's look at the answers. So it's about 60 to 70, minus 60, very important to say the minus 60 to minus 70 millivolts. Now in your textbook it says minus 60, but in other resources it's minus 70, and actually I think it does depend on the neuron, but if you have to pick a value, let's say minus 60, because that's what your textbook says. So maintaining resting potential does require energy. It's largely generated by the sodium-potassium pump. There are other things involved as well, but the sodium-potassium pump does use ATP. The other things that are involved in maintaining resting potential that you need to remember are, first of all, the permeability of the membrane. The membrane is quite permeable to potassium ions, so even though 2 potassium ions are pumped in from the sodium-potassium pump, some of them leak out again, down their concentration gradient, making the inside of the membrane more negative. The second thing is there are large anions present inside the cell that make it slightly more negative. Number 3, no, it won't. Generator potential won't always trigger an action potential, because if it's a small depolarization produced and it's below something called threshold potential, then the action potential will not fire. I'm going to be understanding why that is the case, why it has to get to this sort of cutoff value before an action potential will fire. So let's get into the action potential. First of all, just make sure that you've marked this work from last lesson. So I think I did put the answers on the resource on Teams, but just in case you haven't marked these 3 sheets yet, here they are. Here are the answers. So if you do need to do that, just pause the video now, mark those, and then move on. But we're going to move on and get into the action potential now. So in the action potential, there are some things that occur, some changes to membrane potential. Now they are depolarization, repolarization, and hyperpolarization. Okay, so first of all, let's just write these definitions down. Depolarization is basically the membrane becoming less polarized. Remember, it starts off at minus 60, and I'll use the textbook values, minus 60, and then it gets less polarized. It gets less polarized all the way up to 0, and then it briefly goes positive, up to plus 40, but it's getting less polarized. Then after it becomes less polarized, depolarization, it gets polarized again. It repolarizes. So it returns from the positive back to resting potential, which is negative. But when it does that, it briefly just overshoots. It goes a little bit too negative at the end of repolarization. We call that hyperpolarization, and that's a short-lived event. Okay, let's look at a simulation to kind of focus in on those different terms and to kind of start us thinking about this. It's a PhET simulation. I showed it to you yesterday, but let's have a look at it again. So here we have the membrane of a axon, and down here we can see the membrane potential chart. Now, first of all, if we zoom in, we can see the presence of ion channels here. We've got sodium-gated ion channels. We've got potassium-gated channels, and we've got these leak channels. We're mainly going to be focused on the action of the gated channels today. So let's zoom out a little bit, and first of all, just run the simulation and look at how ions move. Okay, so we're going to look at the charges in the membrane, negative on the inside, positive the outside. So we're looking at how the ions move and how the charge is changing. There we go. Here we go. So sodium ions flood in. That is depolarization, and then potassium ions flood out. That's repolarization. Let's do that again, because I think that was a bit too quick. Slow motion. Let's have the potential chart and then stimulate the neuron. So we should see sodium ions flood in. That's depolarization. Boom, there we go. Look at all those red dots going in. And now look at the green dots going out. Those are potassium ions leaving, repolarizing. So we've got depolarization, repolarization, a little bit hyperpolarized here. It's a bit too negative, and then resting potential will be restored. Okay, so let's see if you can correctly kind of identify those different terms. Here's a quick question for you. Here we go. Pause the video and see if you can do this one. Okay, did you get the answer? So down here we've got resting potential, so we can already kind of rule out some. We can kind of choose A, B, or C, not D. Here we're having depolarization, so 2 is depolarization, so that's, well, it's B, isn't it? Let's just double check. 3 is repolarization, yes it is, and 4 is hyperpolarization, so it's B, yeah, that's right. Okay. Okay. Let's look at this in a little bit more detail now, and let's look at what's happening with the ion channels with a different animation. So, here we go. Okay, a nervous impulse. A nervous impulse is a temporary reversal of the potential difference in the axon, caused by the distribution of ions changing. So if we zoom in, resting potential in the axon is when we have positive on the outside and negative on the inside, an unequal distribution of ions. So because mainly of the sodium potassium pump, we basically have a lot of sodium on the outside of the axon and a lot of potassium on the inside of the axon. So we have concentration gradients in place already. So there are special channels that are called voltage-gated channels. Okay, this is a new form of ion channel that we haven't met before. Now these voltage-gated channels, they will open or close depending on the membrane potential of the membrane around them. So let's look at what happens. When depolarization happens, when the membrane potential, I'll just pause it there just for a second, when the membrane potential gets to that critical threshold value of -50, voltage-gated channels open. These are sodium voltage-gated channels. Look at these ones, these purple ones. They open up, and therefore sodium ions flood into the membrane, down their concentration gradient, which is already there. Okay, so this actually is a passive process itself, but it's dependent on the concentration gradient that's already there. So as they flood in, the inside of the axon is becoming less and less negative and will eventually become positive. So there's a brief reversal of the charge, and the inside of the membrane becomes positive. And here we can see this flipping. Okay, I'm just going to take it back a step there and pause it at the critical value. So we can see this brief reversal of the polarization. Now this wave of depolarization moves along the axon, because as this little bit here becomes positive, those positive charges in the axon there, they kind of spread out a little bit. They leaked sideways, causing the next bit of membrane to become depolarized and the voltage-gated channels to open. So it's a bit like almost like a Mexican wave kind of event. So the action potential propagates along. Now, after about 0.5 milliseconds, the channels close. They kind of have a timer mechanism, basically. So they close after a brief period of opening. And then there's a second type of voltage-gated channel, which is a little bit slower to open, but they are potassium channels. And when they open, after the sodium ones have kind of closed, they allow potassium ions to flood out of the axon, and these repolarize the axons. They allow positive ions moving out of the axon, therefore turning it more negative. So these are responsible for repolarization. So then we have behind the action potential, we get this repolarization and things return to negative. Now because so many potassium ions have left the axon, it briefly sends the membrane potential on the inside a bit more negative than it was. So that is called hyperpolarization. Okay, so that's the overview. And now we're going to make some notes on that. Okay, so you didn't need to make any notes on that. I hope you didn't. Now we're going to make some notes. Here we go. Right. Boom. All right, so writing on this sheet, let's get going. I'm going to ask you to write some notes. So what's happening at stages 1 through 9 of this action potential. And I'm also going to get you to kind of draw some ions on the kind of membrane diagram below. So you might want to get out 2 different colored pens. So maybe you have sodium ions as one color and potassium ions as another color. Let's look at it now. So the first thing, resting state, minus 60 millivolts, more sodium outside and more K plus inside. So here we have the state. I've just kind of revealed in these pictures below. Here we have the state at resting potential. We've got the sodium potassium pump working, and we've got these voltage gated sodium channels are closed. All right, what happens at 2? Well, at 2, sodium channels open. Okay, so sodium diffuses in down the concentration gradient. So the question here is, what's caused these sodium channels to open? Well, this might be this beginning of the action potential here might be caused by a generator potential. So the sodium channels opening here could be channels that open in response to a stimulus in a receptor cell, or it might just be sodium moving down the axon as the axon sort of upstream has kind of already depolarized. Okay, so this is just the initial influx of sodium. At 3, the membrane depolarizes and it reaches the threshold of minus 50 millivolts. This is the threshold value, which means that voltage-gated sodium channels open. So down here, now we have this sodium channel that was closed before, now this one is open here. And maybe we could write at minus 50 millivolts, because that's the kind of point at which they open up. At 4, by the way, pause the video anytime you need, of course. At 4, we have positive feedback. Because voltage-gated sodium channels open quickly, more sodium comes in and the potential difference becomes more positive. So then something quite interesting happens. We have this sort of gated channel here, which basically closes. So this is actually, I've added this in, this is actually what the sodium gated channel is like. You don't have to draw this diagram if you want, but this is kind of like a ball and chain mechanism. So basically when the membrane becomes sufficiently positive on the inside, it kind of pushes this kind of ball out. So that's how the channel actually opens. But then, so this positive feedback causes the potential to reach a peak of about plus 40 millivolts. And then the channel actually closes. So it has kind of like a timer in it. It's a protein that kind of can tell time, I suppose. And it will shut. So the sodium channel shuts and potassium channels then open. So once the potassium channels open, we have potassium ions moving out of the cell, bringing the membrane potential lower again. Repolarizing, and actually they flood out the cell so quickly that actually we briefly have a hyperpolarization, which means that when the cell is hyperpolarized, it could not be stimulated again. Okay, another action potential won't happen there because the cell's kind of like too polarized, basically. Then we, over time, have returned to normal. The sodium potassium pump, over time, kind of gets us back to resting potential. So that was quite a lot of information. So make sure that you've got all that down and that you can kind of run through the steps. So in class, what we normally do probably would be to pair explain this to our neighbor. So what you might want to do is finish writing all these notes and then potentially cover the kind of the boxes along the top with a piece of paper or something, and see if you can just say them back. So see if you can say what happens at each step 1 through 9 of the action potential, and that'll help you learn it. Okay, so pause the video if you want to do that now. And now I'm going to look at one more really good animation of this, just to kind of hopefully solidify that for you. So that animation is here. And stay with me, we're going to watch it together.
You experience when you burn your hand results so quickly in an action by your muscles. Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system.
A relay neuron.
From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection
And we're going to
called
look at this
a synapse.
in another lesson, but I'll let it play out.
The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
Okay. All right, okay, so 2 more things that I'd like us to do, would like you to do. First of all, I'd like you to answer the questions on page 51 of your textbook. So here they are, questions 1 through 6. Read through that page 50 to 51 one more time, I would say, then answer questions 1 to 6. Okay, pause the video whilst you do that, and the answers will come up in a minute. Okay, so here you go. Here are the answers for that. There they are. Mark your work, green pen it. But don't go yet, because I've got one more task for you to do, which will take a bit longer, kind of like a sort of mini homework task for this lesson. So I'd like you to write an essay Probably a 6 marker here, although you could probably say enough for 9 marks. And here's the essay. Explain how action potentials are transmitted along a non-myelinated neuron. Now, don't worry too much about the non-myelinated part. Next lesson, we're going to look at the difference in how an action potential travels in a myelinated versus non-myelinated neuron. And today we've been looking at a non-myelinated neuron. So give this a go. Say at least 6 things, hopefully 9 things. There's some bullet points there on the right. Just to kind of remind you of things you can say, but it will really help you actually to kind of put this into a paragraph or 2. It kind of just really checks to see if you understand it, and if you can write it in a paragraph or 2 and it all makes sense to you, then that means you should know it pretty well. Okay, so that's the second lesson for this week, and then the next lesson that I'll do for you guys coming up will be about saltatory conduction, which is kind of when the action potential jumps along a neuron if it's myelinated. Okay? All right, hope that all makes sense, and I'll see you soon. Bye-bye.